Well, thank you for the introduction, Dr. Carlson. And I have no conflicts of interest to declare. So, as I'm sure everyone knows, some of the variants in the pool of viruses present in an individual during HIV infection carry polymorphisms or escape mutations that allow for abrogation of the cytotoxic T lymphocyte response against HIV-infected cells. And so these viruses are selected for in infected individuals in a manner that's broadly reproducible based on the host HLA class 1 profile. However, upon transmission to an HLA mismatched host, many of these polymorphisms will revert back to the consensus residue at these sites, such that the frequency of these polymorphisms is relatively stable in the population over time. However, some of these mutations may persist upon transmission to an HLA mismatched host, such that they accumulate at the population level. And this is shown in the bottom panel of this figure here, where the pink polymorphism is selected for and subsequently is passed along in transmission, such that it becomes the dominant form of the virus present at the population level. And so that's the objective of this study, to investigate the consequences and extent of HLA-associated polymorphism accumulation in the HIV-1 polymerase gene over the course of the epidemic in North America. We specifically focus on two portions of the polymerase gene, protease and the first 400 codons of reverse transcriptase, and then all of integrase. So in order to do this, we assembled two cohorts of linked HLA class 1 and HIV-1 polymerase data from two time periods in the epidemic. So we have a historic cohort of specimens from 1979 to 1989, and a modern cohort of specimens from 2001 to 2011. Our cohorts are roughly equal in size, with about 300 unique individuals each, and come from 4 key epidemic cities, so Vancouver, Boston, New York, and San Francisco. Our cohorts are well matched for HLA class 1 allele frequency, and the most recent common ancestor of our sequences, which represents the epidemic founder virus, dates to about 1968, which is consistent with the arrival of HIV to North America sometime in the 1960s. So what we observe is a roughly twofold diversification of HIV-1 polymerase between our historic and our modern eras. And this is shown here in the figure with we have two maximum likelihood phylogenetic trees of our historic cohort on the left and the modern cohort on the right. And so what we see is that the modern cohort sequence tree is roughly twofold wider or more expansive than that of the historic cohort. And this indicates a diversifying epidemic. And so when we investigate this diversification, we see that diversifying sites are enriched for HLA-associated sites. And so this suggests that HLA-mediated immune selection pressure is an important driver of the diversification and evolution in HIV-1. So since HLA-mediated immune selection pressure is important, we investigate the frequency of 20 HLA-associated polymorphisms that were identified via phylogenetically corrected statistical methods in individuals who lack the selecting HLA class 1 allele between our historic and our modern cohorts. And so what we see is, on average, we see a 2.6-fold relative increase in the frequency of polymorphisms between our historic and our modern eras. And in general, on average, polymorphisms are increasing in frequency between our historic and our modern eras. Some, however, do remain at stable frequency over time. One to note is that reverse transcriptase codon 135, where HLA-B5101 selects for a T. And so this polymorphism has been hypothesized to be trending towards fixation in a Japanese population, but our data suggests that, at least in North America, this won't be happening anytime soon here. So it would be helpful if we had to investigate this accumulation further if we had sequence data from even earlier in the epidemic, so prior to 1979, which is when our oldest sample is from. We don't have this sequence data or host data, so in order to investigate this, we use phylogenetic reconstruction methods to infer sequences that are even older in the epidemic. And so this is shown in this tree here, which is a time-constrained phylogeny of our historic and our modern cohorts. And all nodes in the tree to the left of the red line indicate inferred reconstructed ancestral sequences that represent sequences from even earlier in the epidemic than we actually have specimens for. So when we investigate the frequency of our HLA-associated polymorphisms in this group of sequences as well, we see that in our reconstructed ancestral sequences, polymorphisms are at lower frequency than either our historic and our modern cohorts. So this lends more support to the idea that as the epidemic progresses, there's this accumulation of HLA-associated polymorphisms over time. We also wondered whether or not the characteristics of a particular HLA class 1 allele might affect the spread of its associated polymorphisms. And specifically, we hypothesized that polymorphisms of protective alleles might be spreading to a greater relative extent than others. And this is in fact what we see. We see a strong, significant inverse correlation between the hazard ratio of progression to AIDS of an allele, so where hazard ratio of progression to AIDS less than 1 indicates protection, and the median fold frequency increase of the polymorphisms associated with that allele. So this suggests that polymorphisms of protective alleles are increasing to a greater relative extent than others, and this could potentially diminish their protective capacity as the epidemic progresses. So this is all fine and good, and we observe this accumulation of polymorphisms over time. But what we really want to know is how this is impacting the ability of hosts to naturally fight HIV infection. And in order to investigate this, we determined pre-adaptation of average circulating sequences in each of our epidemic eras, to determine how pre-adapted they are to people's HLA profiles. And so in order to do this, we consider the HLA profile of an individual and our published list of escape mutations, and investigate which HLA-associated sites are relevant to an individual's HLA class 1 profile. So in this example, there are 5 HLA-associated sites that are relevant to this individual's HLA profile. We then look in an HIV sequence at these sites to see whether or not these sites, how many of these sites carry the escape form. And so in this sample sequence, 2 out of 5 HLA-associated sites carry the escape variant. So we can determine how pre-adapted the average sequence over a whole data set of sequences is to that person's HLA profile by determining this number of escape sites in every sequence in a data set, so say our historic sequences, and giving a median number of escape sites. So the median number of escape sites in this sample data set to that individual's HLA profile is 1 out of 5. So the average circulating sequence in that time period is 20% pre-adapted to the HLA profile of that individual. And we can repeat the procedure with a different data set of sequences, so say our modern sequences. And we see that in this data set, the median proportion of escape sites is 2 out of 5. So the average circulating sequence here is 40% pre-adapted to that individual's HLA profile. Excuse me. So in order to investigate this with our data, we pull the HLA profiles of our historic and our modern cohort and investigate how pre-adapted the average circulating sequence in our most recent common ancestor, our reconstructed ancestral sequences, our historic and our modern cohort, are to their HLA profiles. And so what we see, regardless of era, is that the average circulating sequence is 0% pre-adapted to the HLA profiles of the majority of individuals. And this is shown in the figure here, where the y-axis represents proportion of the population, and the different colors indicate different levels of pre-adaptation. And so regardless of era, the pale purple color dominates, and the pale purple color represents 0% pre-adaptation. So this is a good thing. But what we do see over time is that there's an increasing proportion of individuals who have experience higher levels of pre-adaptation in that average circulating sequence to their HLA profile. So pre-adaptation remains low, but is increasing over the course of the epidemic in North America. So in conclusion, the HIV-1 polymerase gene has diversified roughly twofold since 1979 in North America. We see a 2.6-fold relative increase in polymorphism frequency between historic and modern eras. The average modern circulating sequence remains 0% pre-adapted to the majority of hosts, but there is increasing levels of adaptation over time. So acknowledgements from SFU, my wonderful supervisor, Dr. Sabrina Brumet, Daniel McMillan, who developed some of the web tools that made analyzing this data much, much easier, Onlay and Laura Cotton, who collected some of the sequence data used here, and other members of the Brumet and Brockman labs. From Microsoft Research, Dr. Jonathan Carlson, who's also chairing our session today, and who identified HLA-associated polymorphisms for us. From the British Columbia Center for Excellence in HIV/AIDS, Dr. Art Poon, Rosemary McCloskey, and Richard Liang, who performed ancestral reconstruction and epidemic dating. And our colleagues from across North America who provide access to samples without which this study would not be possible. And finally, to the conference organizers for allowing me to speak today, and also for sponsoring my attendance. Thanks.